So next topic of sexual reproduction in flowering plant is structure of stamen. Okay, the first I will draw the structure of stamen. The stamen is like that. It has two lobes. You can see that's called as anther. Okay, and you can see this is this portion that's called as connective, and you can see this small thread like structure that's called as filament. Okay, and this is the structure of stamen. So now we will see this actual structure of stamen. So androecium, as we know that androecium is a male reproductive floral whorl. It's having definite number of individual members, that's called as steamen. Okay, so whenever we study that, this is nothing but the male reproductive part of the flower, right? So androecium, we know that. Okay, in previous lecture we will see all the whorls of the flower. So androecium is this male reproductive whorl. Okay, reproductive floral, we can say like that, floral whorl, that having a definite number. Okay, the number of this stamen is not fixed, not always 1, 2, 3. Okay, so having a definite number of individual members of this family, that's called as stamen. So I can say like that, this androecium is a male reproductive whorl having definite number of individual members. That members is known as stamen. Okay, now what exactly? So this you can stamen. The stamen contain 3 parts: anther, connective, and filament. So it consists of filament, it consists of anther and connective. This portion is known as anther, this portion is known as connective, and you can see this portion that's called as filament. So you can see this filament, it's very basal, sterile and slender stalk, right? So it is, what is filament? Filament is nothing but a basal. Okay, it's present below that, or it's a basal, it is sterile. This part is not fertile, it's sterile and slender stalk. And anther, you can see it is an upper sac, but it is a fertile part of the stamen, and it is a non-fertile plant. That's why it is called as sterile plant. It is sterile part, and this is called as fertile part. Okay, so this is anther, and where is anther? So it is upward. You can see the sac-like part. It is upward sac-like fertile part is there. This is a fertile part is there. Then generally you can see this anther, right? This anther consists of 2 lobes. This is a 1 lobe and this is 2 lobes. Okay, so anther consists of 2 lobes. 2 lobes. Okay, this lobes is known as yes, you can see this 2 lobes. Okay, but sometimes it contain 1 lobe also in some species of the plant, right? So generally the anther consists of two lobes, but sometimes anther consists of one lobe. And each anther lobes, this is one, this is two, and each anther lobe, you can see, has two pollen sac. It contains two pollen sac. So if I am talking about two lobes, so how many pollen sac is there? 1, 2, 3, 4, 4 pollen sac is there. Okay. So here I am writing each anther lobe has two pollen sacs, or that pollen sac is also known as pollen chambers. Okay, and this pollen chambers is also known as theca. Okay, it means that each anther is a dithecious. It means they have two pollen chamber in each anther lobe, so they are dithecious. Okay, and that pollen chamber is also known as pollen sac, or is also known as theca. Okay, so these two anther, you can see the joining of these two anther lobe is a strip of sterile tissue. You can see here this strip. Okay, I use another pen so you can see. Okay, this portion जो मैं ब्लू कलर से कर रही हूँ, right? Fine. Okay. So the joining of these two anther lobes is nothing. Is a strip of sterile tissue, and that is called as connective. So what is this connective? It's nothing but it is a strip of sterile tissue, and this sterile tissue helps to join the two anther lobes. Anther is four-sided structure, consists of 4 microsporangia. Okay, you can see this is the structure of anther, right? So anther is a four-sided structure. Okay, that consists of 1, 2, 3, 4 microsporangia and 2 sporangia in each lobe. So large number of pollen grains are produced in a anther lobe. Understood? So here it contain, how many contain? It contain 4 microsporangia. Okay, and 2 microsporangia in each lobe. You can see. Okay, this is a one lobe. Okay, it contains 1 microsporangia, 2 microsporangia, and it means that each lobe contains how many microsporangia? 2. If I am talking about it, 2 lobes are there, how many microsporangia are there? 4 microsporangia, and inside this microsporangia, large number of pollen grains are present. So you can see that large number of pollen grains are produced inside the anther lobe. Yes. So here the large number of pollen grains, right, are produced inside the anther lobe. Then what is this function? Okay, what is the function of stamen? What is the necessity of this structure in the flower? First is that, what is the function? It produces the pollen grain. What? It produces pollen grains. Second, this pollen grains develop into the male gametophyte with the 2 male gametes. Okay, so these are the 2 functions of this structure, that's called as stamen. Okay, now we will see the internal structure of that, or now we can see the structure of Microsporangia. Okay, how it's look, that is, or in other we can say like that, the tiers of anther. Okay, so internally anther shows so many internally this anther, you can see this is anther, so this shows this anther wall along with this pollen sac. Okay, so now next we will see the structure of Microsporangia. Okay, so next we will see the structure of Microsporangia. Okay, or this is also known as TS transverse sections of anther. Okay, in exam it may be asked the structure of Microsporangia or instead of TS of anther. Both the answers is same. Okay, first I will draw the structure of Microsporangia, right? So here I am drawing, I will draw okay. Again it has one more membrane, so I will draw one more layer like that. Then there are 4 Microsporangia is there. Okay, that is nothing but a pollen sac. Okay, how many pollen sac is present in anther? If that 2 anther lobe is there, the 4 pollen sac. So each lobe contains 2 pollen sac. Okay, now this is another layer, that is called as okay. Now see, you can see this outermost layer, that is called as Epidermis. Then second layer you can see this, that is called as Endothelium. And this layer you can see, that's called as middle layer. And this you can see, this layer, that's called as septum. Okay, this is actually the structure of pollen, TS of this anther, or structure of Microsporangia. Okay, now we will discuss. So you can see internally, we internally see the structure of Microsporangium, right? So internally this anther shows the anther wall. It contains anther wall and pollen sac. You can see this layer. So this is nothing but anther wall, and you can see this 4 round structure, that rounded structure, that is called as pollen sac. So I can say like that, internally this anther shows anther wall and pollen sac. Okay, now first we will see this anther wall. So in this anther wall is divided into four layers. First is called as epidermis. Okay. Second is called as endothelium. Third is called as middle layer, and fourth and the last layer is known as septum. Okay, so these are four layers present in anther wall. So we will see one by one. So first we will see the epidermis. Okay, you can see this layer, outer layer, that is called as epidermis, and it is a outermost layer. It is a outermost protective layer. Okay, this epidermis is the outermost protective layer of anther, which consists of flattened cell, cells. Okay, so first layer of anther wall is the epidermis, and this epidermis is the outermost protective layer of this anther, which consists of flattened cells. Second layer we will see, that is called as endothelium. You can see this layer, that is called as endothelium. So you can, I wrote this. Okay, now we will discuss with the endothelium. So you can see endothelium is present below the epidermis, right? So it is present below the epidermis. Right? Next, the cell of this endothelium, you can see the cell of this endothelium shows a fibrous thickening of callus. It shows fibrous thickening of callus. Okay, so fibrous thickening and hygroscopic nature of these cells that help in dehiscence of the anther at maturity, right? Because of this fibrous thickening. Okay, we are talking about this second layer of anther wall, that is called as endothelium, right? So endothelium, where it present below the epidermis, right? And the cell of endothelium shows fibrous thickening of callus. Okay, and this fibrous thickening of callus along with this hygroscopic nature of cell. Okay, so this fibrous thickening and hygroscopic nature of these cells helps in dehiscence of anther, but at maturity. Okay, so what do you mean by hygroscopic nature? What do you mean by hygroscopic nature? Yes, anyone know? What do you mean by hygroscopic? Camphor, you know, it is a hygroscopic. Sodium, you know, it is a very high. Camphor यानी कपूर जिसे हम कहते हैं. Okay, it is hygroscopic. So because of this hygroscopic nature of this cell, so what will happen? The cells will become dry, and because of this dryness of these cells and this fibrous thickening, what will happen? It result into the dehiscence, यानी breakdown of the anthers and removal of lots of pollen grain from that. But this occurs at the maturity level. Once it's matured, then dehiscence of anther occurs. Fine. Next layer we will see, that is the third layer, that is called as middle layer. So this middle layer, it's present. You can see this one. This middle layer is present inner to the endothelium. Yes, it is present inner to endothelium, and one to two layers of this parenchyma cells are present surrounding the sporangious tissues of Microsporangia, and this type of cells is degenerate at the maturity, means after the formation of a microspores, right? So what is this middle layer is present inner to this endothelium. It is contain 1 to 3 layers of parenchyma cell. What is parenchyma cell? Cells are present, and that cells are present surrounding the sporangious tissue. Where it's present? It is surrounding of sporogenous tissue of Microsporangium. Okay, so where is the sporangious? Where is the sporogenous tissue? So you can see inside that this layer is a tapetum, and inside that tissues are there, cells are there. That cells is known as sporogenous cells or tissues. Understood? So each microsporangium contain at the center of each microsporangium one specialized cells is present, that is called as sporogenous cells. Okay, so here in middle layer you can see this layer. Okay, this layer. So this layer, middle layer, is present inner to the endothelium, and in this layer 1 to 3 layers of parenchyma cells are present, and that is surrounded by this one of the special cells that's called as sporangious tissue of micro. These cells of this layer, okay, whatever the cells are present in this middle layer degenerate at a time of maturity. Okay, yani at a time of maturity, it means what it means when it's become mature after the formation of microspore. Okay, what do you mean by maturity? Maturity is nothing but the formation of microspore. Okay, so here this middle layer get degenerated once they form a microspore. Okay, so next layer, fourth and last innermost layer we will see, that's called as tapetum, right? So tapetum is a very important layer present in anther wall. It is the innermost layer of the anther wall. Fourth layer, it's called as tapetum, right? It is the innermost layer. You can see this layer. I will color by another. Okay, जिस layer को मैं blue color दे रही हूँ, okay, that is called as tapetum. So the tapetum is innermost layer of anther wall. Innermost layer of, it is layer. It's the innermost layer of anther wall, and it is present around the pollen sac. You can see this is a pollen sac, so it is surrounded by the tapetum. So it is the innermost layer, and it's present around the pollen sac. Okay, what is the need of this layer? Why this layer is very, very important? Because it provides nutrition to the developing pollen grain. Whatever the pollen grain are present inside this pollen sac, right? So this steptum provides a nutrient. It provides nutrition. It provides nutrition to the developing pollen grain. Okay. It also contribute in the formation of this layer, steptum layer. Steptum layer is also contribute in this formation of that sporopollenin, a component of pollen exime. Okay, this layer play a very significant role in the formation of sporopollenin. Okay, and that sporopollenin is nothing but the component of pollen exime. What do you mean by pollen exime? This is the two wall present in pollen. Outer wall is known as exime, inner wall is called as intine. Okay, so this sporopollen is one of the component of the of this outer layer of the pollen, that is called as exime. So we will see what is this. So what is the need of this sporopollenin? This give more resistance compound. It is biologically most resistance compound. Okay, so it resists the physical, biological and chemical attack external. It's prevent the pollen grain. So you know the steptum, right? So steptum, or already I told you, steptum play a significance, or it may a large contribution in the formation of the pollen exime. So I can say this steptum is glandular in nature. Okay, this steptum is what is the nature? It has two types. Later on we will see. First, this steptum have a glandular nature and its form cubish bodies. Okay, and this bodies transports, and this cubish bodies transport sporopollen. Okay, to exime. Why these bodies transport sporopollenin to the exime, that is the outermost layer of this pollen grain? Okay, because to resist physical, biological and chemical attacks. Okay, so this sporopollen plays a significant role to fight against physical, biological and chemical attacks. Okay, that is a significant compound present in the pollen exime, that is a layer of pollen grain. Okay, now you got it that much. Now we'll see the steptum basically has two types. Okay, the steptum has two types. One is called as secretory or glandular steptum, and second it's called as amoeboidal, or it's called as periplasmodial. It's also called as periplasmodial. Okay, so this is innermost layer, that is called as steptum. has two types, secretory glandular and it's amoeboidal, or it's also known as periplasmodial septum. So what is the role of here secretory or we can say glandular septum? This septal cells present, okay, whatever the septal cells present in that layer, so this septal cell remains in situ all through this development of microspores, and finally they degenerate. Okay, so in this type of septum, what will happen? The septal cells is remains in situ, अंदर ही रहते हैं for all development of the microspore, and once the microspore develop, it's finally degenerate. Okay, this is occur in secretory or glandular type of septum. Second, it's called as amoeboidal, or it's also known as periplasmodial septum. So here the septal cells breaks through the radial wall to release the protoplast. Okay, वो क्या करते हैं? What happen in amoeboidal cell? Whatever the cells is there, septal cells, वो क्या हो जाते हैं? Break करते हैं. क्या break करते हैं? It's break the wall, radial wall. Okay, and to release protoplast. And to release protoplast. कहाँ पे रिलीज करते हैं प्रोटोप्लास्ट? प्रोटोप्लास्ट इन पोलन chamber. Okay, and now this protoplast fused and to form a periplasmodial like structure. So that, or they form a periplasmodium. So that's why this type of structure, this type of septum is known as amoeboidal or periplasmodial septum. Understood or not? Okay. So that is about the 4 important layer of anther wall. Okay. After this 4 important layer of anther wall, we will study the sporogenous cells. Okay, we already know the sporangial cells, right? Sporangial cells are present inside the septum. Okay, now this sporogenous cell, or it's also known as tissues. Okay, this sporogenous cells, it is compactly arranged. It's compactly arranged homogeneous cells which are present at the center. You can see at the center of each microsporangium when they are young. Okay, whenever they are young, microsporangium. So this sporangium cells, okay, it arranged compactly. This compactly arranged by homogeneous cells. Okay, and it is present at the centers. Okay, when the microsporangiums are in young stage. Fine. The next part, it's a pollen sac. We will see. First is pollen anther wall. Second part is called as pollen sac. That pollen sac is also known as Microsporangium. Okay. Now we see. As we know that anther is a bilobe structure. Okay. Here again I will draw the line structure. This is anther. Okay. So you know anther is a bilobe structure, and each lobe contains 2 sporangia, 1 and 2. Here 1 and 2. This is one lobe. This is second lobe. Okay, so each lobe contain, here I am writing, each lobe contain 2 sporangia, or instead of sporangia we will call as pollen sac. Okay, here the pollen sac and sporangia both are same thing. Okay, is the alternative name of that. So each lobe contain 2 sporangia, that is nothing but a pollen sac. At 4 corner 1, 2, 3, 4. Okay, at 4 corners 4 microsporangia is there, and this pollen sac contain the number of diploid sporogenous cells. You can see each sporangia contain each we can say this pollen sac. Okay, this is pollen sac, right? Now this pollen sacs contain number of sporogenous cells. That sporogenous cells is also known as microsporocytes. Okay, so what I'm just, you know that each lobe is there and each lobe contain 2 sporangia. So 2 lobes contain 1, 2, 3, 4 sporangia at 4 corners, and this pollen sac, the sporangia, contain number of, you can say inside, I will draw a circular shape structure, that is called as sporogenous cells. That sporogenous cells is also known as microsporocytes. Okay, this microsporocytes is divide mitotically. Okay, mitosis occurs, is divide mitotically to form microspore mother cell. To form microspore mother cell. Okay, and this each diploid microspore mother cell, it is diploid in nature. It means they contain 46 number of chromosomes. Okay, so this microspore mother cells undergoes meiotic division there. It undergoes meiosis. This microspore mother cells undergoes meiotic division to form 4 haploid microspore. These are diploids. After meiosis they form a haploid microspore, or this haploid microspore is nothing but we can call as pollen grain. Understood? Okay, again I will repeat this section, that is called as pollen sac. Okay, so this pollen sac is also known as microsporangium. Okay, this portion is nothing but a pollen sac. This pollen sac is also called as microsporangium. As we know, this anthid is a bilobe structure, and each lobe contain how many sporangia? 2 sporangia, or 2 pollen sac is there. Okay, it means 2 anther contain 4 microsporangia at 4 corners. Yes. Now this pollen sac contain number of, you can see here, it's a number of diploid sporogenous cells, and that sporogenous cells is also known as microsporocytes. So these microsporocytes which divide mitotically, okay, means it undergoes mitosis division to form microspore mother cells, or it's a pollen mother cells. Okay, it is also known as microspore mother cells, or it's also known as pollen mother cells. Okay, that they are diploid in nature. So after the microsporocytes which divide mitotically to form a microspore mother cells, and each diploid microspore mother cells undergoes meiosis, or undergoes mitotic division to form 4 haploid microspores. Okay, and that haploid microspore is nothing but the pollen grain. So this is actually the structure of Microsporangia. Okay, thank you.